general. I think you'll enjoy the last part more, but you might like the first part too. It's all kind of interesting. Nervous systems and sensory and motor mechanisms. You can see why I combine these two into one chapter. There's a lot of good stuff in both these chapters. I'm going to treat it as one chapter on the exam. It's not like this section is going to have a lot more questions to it. It won't. Go by the study guide. Also refer to the email I sent you this morning telling you what's going to be on the quiz. Okay, first up, neurons. Not to be confused with nephrons. I don't name this stuff, I just teach it. Okay? So be aware that these are very similar sounding names, distinguish between them. Nerve cells. Estimated about 1 billion per brain. That's just an estimate, an average. Some of you might have more than that. I'm not going to do that. Specialized circuits dedicated to specific tasks. Your brain is not like just this generic mass of gelatinous material inside your skull. It is divided into sections. If you want to really get into the breakdown of the brain, take an anatomy and physiology course, because then you can see what different parts of the brain, what regions deal with different parts of your thinking, of your abilities, physical, mental, whatever. Ganglia are clusters of neurons, and we've seen some of these in lab. The green are the neurons, and the blue would be the nucleus. So neurons are nerve cells, ganglia are clusters of neurons. Nerve net-like pattern of nerves running throughout an animal's body. Some nerve nets are very sensitive, like ours, very specialized, very sensitive. Others, not so much so. You can go back to a hydra, something that really doesn't even have a brain per se, but it has this nerve net. It allows it to sense what's going on in its environment. It puts its tentacles out looking for food, and when something touches it, the tentacles grab it, they stun it, and they bring it into the mouth. So it does have basic reactive systems going on. So it can react and respond to its environment. A sea star has a very basic nerve net, basically a nerve ring and then a radial nerve going down each arm. The planarian that we talked about before, the flatworm. A leech, a little more advanced than the planarian. Arthropods, you start to see a concentration here and in the leech of a brain. Not so much here. You have eye spots, a little bit of a brain, and then of course you've got the chemical sensors that you use. Chitin, it's a type of mollusk. Squid's another type of mollusk, actually a little more advanced. Anybody ever heard of a chitin before? So I've got some in the lab if you're really dying to see what they look like. Let's go with the squid. We're getting more advanced here, especially with the eyes of the squid. Very advanced for an invertebrate, very advanced for a cephalopod. And then you get to the vertebrates where you definitely have more of a concentration of a brain with cephalization, with sensory organs mostly concentrated in the front of the face in the anterior region of the organism, and then the nerve net that extends beyond that. Okay, the next three kind of go together, and they talk about how the nervous system works. First, you've got the sensory neurons. Transmit information from receptors to detect external stimuli and internal conditions. External could be light, sound, temperature, touch, taste, smell, 5 senses. Internal, you've got these sensors running all over the inside of your body. Blood pressure, making sure that your blood pressure is what it's supposed to be, that it's not too high, not too low. Blood pH, your blood pH is very sensitive. It's got to hang around about 7.4. Too high or too low, 7.5, 7.3. Anything above or below that could kill you. But you've got tons of buffers in your blood to make sure, and with the sensory neurons working in conjunction with all these buffers, send it where it needs to go, so that your blood stays right at where it's supposed to, around 7.4. Blood carbon dioxide levels, making sure you don't have too much CO2 in your blood, that your lungs are working efficiently enough to carry oxygen to all parts of your body and to pick up and remove the carbon dioxide, and muscle tension. You've got a lot of nerves in your muscles to control your muscles, to make them move. But you've also got nerves there that sense when they do move. It kind of gives you this orientation, sense of where you are in time and space. Whether you're standing or sitting, whether you're running or walking. sensory neurons in Drosophila, olfactory and gustatory. We'll talk more about this. What's olfactory? Smell. What's gustatory? Taste. Check it out. It's got where you would expect gustatory taste around the mouth. But look, it has it on its legs too. So when a fly lands on your food, it's not trying to eat your food, it's tasting. Make it any better. Taking it probably 5 minutes before it was on some of that dog urine or cat urine or something like that in the yard.
Don't they throw up on the food though?
Do what?
Don't they throw up on the food?
In order for them to digest food, they will release digestive enzymes and then kind of slurp it back up. So, yeah. So sensory neurons. Interneurons integrate sensory input, considering immediate input while comparing it to past events. These are the site of greatest complexity of neural circuits. So types of neurons. Here are my interneurons right here. The motor neuron, you see that it's not quite as extensive as the interneurons. The sensory neurons, again, not quite as extensive. So, I think I've given you this analogy before. The light bulb just burns out. You go to change it, you touch it. You're thinking it's out, but you don't realize it's hot. And so as soon as your fingers touch the light bulb, you have pain sensors in your hand, you have temperature sensors in your hand that send a signal to your brain. So your brain starts processing this and thinks, wait a second now. The idiot did it last month. Second, almost third-degree burns to the hand. He didn't let go of it fast enough. So let's see. To resolve this, we need to let go of that light right now. So it sends a signal back down your arm to tell your hands to release and pull back your arm. Release and pull back, release and pull back, release and pull back. The beauty of this is your nervous system works in fractions of a fraction of a second. And before you realize it, ow, that's hot, you're already releasing and pulling back. Because your brain's already figured out that you did, that you shouldn't touch it in the first place. Now you need to let it go as soon as possible. So that's the sensory neurons, the interneurons that process the information, and then the motor neurons transmit information to effector cells, which could be either muscle or endocrine cells. In this case, muscle cells in the arm. So even though this is under voluntary control, you move in your arm. You move your arm to the light bulb, take it out. Sometimes your brain overrides the voluntary control and says, oh, excuse me a second, I need to borrow this, pull it back. And it pulls it back. And that's the specific response it carries out. So neuromuscular junction. The blue are the muscle fibers. The red is the nerve fiber. And these yellow extensions of the nerve fiber are attached to the muscle. And that's what controls the muscle. So you've got 3 neuron process. The sensory that detects things, the interneuron that thinks about and decides things, and the motor neuron that carries out the response, the decision the interneurons make. Make sense? So effector cells, muscle and endocrine cells, we'll talk more about those. The neuron, one of the things I want you to know is the structure of a neuron. If I give you something that looks like this, can you label the parts that are square, or these parts down here? So for instance, I've got dendrites, cell bodies, the axon hillock, the axon, myelin sheathing, and the synapse. Let's start with the cell body. This is the main part of the neuron. This is where you're going to find most of the cytoplasm, the organelles, the nucleus. It's what we think of when we think of a cell. The dendrites and axons are extensions out of the cell body. You will have numerous short dendrites, and they're going to be picking up the signal, because the signal is one way in a neuron. It's going to come in this way, close to the cell body, and then out this way. You'll have one axon that's very long, moving away from the neuron. So the signal comes into the dendrites, through the cell body. One part of the cell body forms this triangular axon hillock that leads into the axon. And this is the way the signal goes. The myelin sheathing is insulation. It's what helps maintain a good signal. When I first had moved into my house, I had been told that there was a rat problem. And the people who lived there before me were basically kids. So I found rat traps everywhere, thinking, okay, never saw any rats, but the damage was already done. And I had to have cable wire rerun because they got inside the walls and chewed away the insulation from around the cable wire. So what was happening is my signal was jumping between two different wires. So I wasn't getting a good clean signal. But the cable wire, with the insulation, I get a clean signal. Same thing with this. The myelin sheathing gives you a good clean signal. When that starts to break down, we'll talk about how and when that happens, you start to lose your signal. Meaning you don't get a nice, crisp response, and muscles don't work like they're supposed to. The synapse is the point that actually is between two neurons, or between a neuron and an effector cell, between a muscle or a gland. And it looks like it's touching here, but actually there's a gap where the synaptic end of one neuron comes close to but never really touches the synaptic end of another neuron. And they call that the synaptic gap or the synaptic cleft. There's a space there. We'll talk about how neurons bridge the gap. And so when they talk about a presynaptic cell, it's a cell before the gap, the synapse, and the postsynaptic cell is a cell afterwards. So make sure you know these 1, 2, 3, 4, 5, 6 points. Glia cells. Support cells essential for structural integrity of the nervous system and normal functioning of neurons. They tend to be 10 to 50 times more numerous than neurons. I gave you the analogy in lab when you were looking at neurons under the scope. I told you you're going to see a lot of little purple dots in there. Those are the glial cells or the support cells. And I gave the analogy of kind of like a stock car. The neuron is like the stock car. Now it doesn't pull into the pit, one guy runs out and takes care of it. There's a bunch of guys, and girls sometimes, who run out and take care of it. Maybe 12 people at one time. Changing the tires, checking the oil, filling the gas, checking the driver of the car, making sure he's okay. Wiping off the windshield, do whatever needs to be done. And the car is maintained and ready to go on its way. Well, these are like your pit crew for your neurons. They take care of the neuron, maintaining the integrity of the neuron. You see around this one neuron, you've got the dendrites, the cell body, the axon hillock, the axon, and around that oligodendrocytes. Two different types of glial cells. You don't need to know that, but just know that they're different types to help maintain the integrity of the cell. The blue is a neuron, the yellow are the glial cells taking care of the neuron. Now there is one specific type of glial cell I will want you to know. So it makes sense you have more glia cells than neurons if you're going to have them taking care of the neuron. The one type I want you to know are the Schwann cells. The Schwann cells are support cells essential for insulating axons of neurons with a myelin sheath, mostly comprised of lipids. And we saw this before in the previous diagram of the neuron. You see they're like rolls of paper towels or rolls of toilet paper just wrapped around us. They start here and they just keep wrapping and wrapping and wrapping. Here's the nucleus of the Schwann cell. And you see them spaced out. We'll talk about why they're spaced too, why they're not just continuous all the way down the length of the axon. But this is the insulation. This is what helps to maintain the integrity of the axon signal, of the nerve signal.
Those are what the myelin are made up of?
Lipids?
Yeah.
Fats?
The Schwann cells?
I mean, it's a cell type that, think about an adipose cell that's used to just wrap around something. That's basically what it is, just full of lipids that it helps to insulate. I mean, it's like when you have wire, you wrap it with like a plastic rubber. So then it's kind of, it has a little bit of flexibility to it, but it doesn't really help the signal to go through. It just keeps the signal in.
That's
Same
part
thing
of
for
why
these. This is just to keep the signal in.
it's for the flexibility, right? The reason why there's the junctions.
Not exactly. We'll get to that. Yes.
So is it the myelin sheath, or is it's the stuff that's myelin sheath is made up of?
It's one and the same. They're made up of lipids, but the myelin sheath are these cells that go, well, the Schwann cells. The myelin sheathing is what, yeah, I see what you're saying. Schwann cells, the myelin sheathing is this structure around all of this. It's mostly lipids. So the myelin sheathing is a part of the Schwann cell.
That's what I was wondering.
Okay, yeah.
Multiple sclerosis. You notice it's a health note. Disease in which the myelin sheath deteriorates, resulting in progressive loss of body function due to the disruption of nerve signal transmission. With a healthy myelin sheathing around your axon, the nerve signal travels very nicely, but what happens is the myelin sheathing starts to break down. Really not sure why or what causes MS or multiple sclerosis, but it is a progressively degenerative disease, meaning once it starts, there's no cure. It will just get worse and worse. It might start that you might have trouble grasping things or walking, and it could lead all the way to where you can no longer walk, and to where you're pretty much bedridden, and to pretty much where you lose almost all control because it just eats away at all the myelin sheathing.
Is that something you can only be born with, or can it have, like, can it start?
Because they're not really sure what causes it, there could be a genetic predisposition that some people, maybe if somebody in the family had it, they might develop it as well. It could be lifestyle, it could be something you contract from the environment, they're just not really sure. So disease in which the myelin sheath deteriorates, you're breaking down the insulation, so you're breaking down the signal, resulting in the progressive loss of body function and the disruption of nerve signal transmission. Okay, let's take a break before we get into this. It's all italics, you don't have to write it down. Be back here at 10:30. Don't be. I'm not going to spend a lot of time with. We could kill this anatomy and physiology course, but we're not going to do it now to just kill you. As ion channels open and close, membrane potential of the cell membrane can change dramatically. When channels open, ion movement is sensitive to electrochemical gradients. Ions move down chemical gradients from areas of higher to lower concentration. That's just a basic principle of diffusion or osmosis. From an area of higher concentration to an area of lower concentration. Ions move along the electrical gradient towards areas of opposite electrical charge. Yes, it's true. Opposites do attract. So in this, it's kind of like a busy picture, but you see sodium and the little round yellow ones. Potassium are the orange diamond-shaped ones. I've got a sodium-potassium pump here. I've got potassium channels, sodium channels. Potassium and sodium channels are pretty much open all the time. And they just let potassium and sodium flow through constantly. They follow the rules of chemicals moving from an area of higher to lower concentration. The sodium-potassium pump is an active pump that pumps against the concentration gradient. It's salmon swimming upstream. Because what happens is, when a signal comes through, it causes the polarity of the cell membrane of the nerve to change. Now, if you were just to wait to let that reset itself, it could take a while. What you want to do, as soon as that signal passes, is to go back to the original polarity. And the sodium-potassium pump ensures that that happens. Otherwise, let's say you touch something hot, you pull your hand back, you still feel the heat in your hand even though you're no longer touching anything hot. Well, what it does is resets the sensing of the initial stimulus, trying to get it back to normal as quickly as possible so you're ready for the next stimulus. So the sodium-potassium pump sees that, ensures that ion movement is regulated by resetting the initial charge. Yes?
Do they, I guess, I don't know to call them, do they know the difference between like a strong stimulus and a weak stimulus? Or is it like is it the same reaction for both?
They can sense it, one, by the incorporation of how many different sensory neurons pick up on it, and because of the intensity of the stimulus. And I'll show you that in a second.
Two questions. One, do they do that because they just aren't trying to get back to homeostasis, so they
Yes.
get shocked and then they try to go back?
Yes.
And then also, salt conducts electricity, right?
I believe so, yes.
Does it? Is that why it can?
The sodium-potassium pump.
Yeah.
It's the ions of sodium potassium that cause this positive ions. And then what happens is, it's with these ions that determines the polarity of the membrane. And so everything's in the currency of these ions is both positive. When the current comes through, it flips those. And so you have to reset them.
It's like an on-off switch.
Yes, exactly. Now I'll give you a little more detail about it in a second. So it's not sinking in yet. Give it a second. Yes.
But that potassium, the sodium actually, is what helps conduct those nerve pulses.
Both sodium and potassium. We'll get into that more in a second. Okay, one of the things I want you to be familiar with are the parts of an action potential. If you look at the stimulus going through your nervous system, say from touching something hot up your arm to your brain, it's like a wave that moves through, starting at your fingers and rolls up your arm into your brain. And then right behind it is this resetting, this reestablishment of normalcy, so you're ready for the next stimulus, and a second wave rolls through. Membrane potential is the electrical voltage across a neuron's membrane, which is measured at minus 70 millivolts. Don't worry about remembering that, just know what it is. I won't test you specifically on that. This is when the cell is not transmitting a signal or simply resting. It's also known as the resting potential. It's right here. So you're just laying there in bed asleep at night. This is your resting potential. You're not doing anything. You're just calm and relaxed. So you're sitting there, you're not picking up any stimulus of any way, shape, or form. It's like watching Keeping Up with the Kardashians, you know? There's no brain activity whatsoever.
Does this relate to like when you have like your foot fall asleep or like when your hand falls asleep?
That's something else. That's cutting off circulation to those areas. But yeah, I mean, it does tingle a lot, it becomes very hypersensitive.
Because of
It's more circulation, but obviously there's a nervous component there too, and it's kind of trying to reset you as quickly as possible.
okay.
That's why it feels kind of funny. The action potential is a rapid change in the membrane potential of an excitable cell caused by a stimulus. Okay, you're laying in bed at night, and all of a sudden you hear a loud noise. And that causes you to jump up in bed. The noise is the stimulus. Now you're up, you're alert. Everything is firing. Now you've got an action potential. So the noise was the stimulus causing you to jump up in bed. So the stimulus, boom, rises. And it must pass the threshold minus 55 millivolts. So, you were asking about the intensity of the stimulus. If the stimulus isn't strong enough to go past 55 millivolts, it just keeps misfiring basically. The stimulus has to be strong enough to pass the threshold to create a change in the cell membrane. Does that make sense? That's your question. And again, minus 55 millivolts, I just put that out there so you can appreciate in comparison and reference to the minus 70 millivolts of the rest. So it has to increase at least by 15 millivolts, usually more than that.
Are you gonna cover the undershoot, like where it goes? Oh, why does it go?
I can touch on that for a second, yeah. But we're just kind of starting right here now. So we came along, boom, we've got the action potential, so we just passed this, we're on the way up to the peak. Threshold is the membrane voltage a stimulus must reach for an action potential to be generated. So I'm coming along, 1 didn't get it, 2 didn't get it, all of a sudden a noise loud enough woke me up, creating the action potential, but has to pass this threshold of 55. Now you might have noticed I changed pictures. I like that diagram, it's pretty colorful, pretty easy to see. On this one though, the peak is, it's no longer called the peak, it's the action potential. And you see that the terms are no longer rising and falling, but we've got depolarization and repolarization. The depolarization is an increase in magnitude of an action potential. What that's talking about is depolarizing the cell membrane. It's causing it to flip from positive to negative to positive. The repolarization resets that, puts it back to normal. So as this wave passes through, almost as soon as it depolarizes, it repolarizes. So repolarization is a decrease in magnitude of an action potential. Now sometimes when they repolarize, and you're asking about this, what do they call it over here? The undershoot, the refractory period, same thing. what happens is sometimes it goes down below this in resetting, but then it resumes back to the resting state. It's just a little bit of overcompensation. Making sure that you get back to the resting state, but then bringing it up to where it needs to be. Armand, question?
Oh, no, I'm amazed that this all happens. It goes back to normal within like 4 milliseconds.
That was in my next question. I was gonna ask that. If there's a lot of stress and say something happened and all of a sudden and it's going on for a while, can it sometimes stay up that high? Where it doesn't reset right away?
That gets into a lot more detail than what we're going to talk about in here, but yes.
Yeah. I would
You
think
can
so.
have repetitive peaks, just one right after the other, depending upon the stimulus.
So it kind of does go down, but then it just pops right back up again?
Right, and there's sometimes the peaks feed into each other and it increases the stimulus. So there's an additive effect to it. And sometimes, so it just depends on all the
The
things coming
situation and everything, okay.
together. Yeah. It's like you go to a concert, but you still get the ringing in your ears afterwards, okay? That's the stimulus, overstimulated and still trying to reset itself.
Could that-
doing.
Could that be like in like another field, like could that be like PTSD or something?
That's getting into more than around with the nervous system. It's possible from the sensory to the interneurons and back to the motor neurons. Yeah.
Does the distance between the myelin sheath have anything to do with like acquired memory or like how quickly you can retrieve the thought?
I don't know if it does or not. My initial reaction to that would probably be no. I think it's more the ability of the interneurons to process information, to make the decision, and then to send out the response, more so than the signal traveling to or from.
What makes like an interneuron better at that or worse at that?
I don't know. Off the top of my head, I would say genetics might help. Getting plenty of rest, the right nutrition, avoiding excessive use of drugs, alcohol, things like that. Anything that would be detrimental.
Would that
B12, can they be developed over a lifetime or are they like
According to some of the commercials I see on TV, yes, there are calisthenics you can do for your brain. I think you can develop if you focus on things to make yourself more aware, better at stuff. Some people are naturally better at stuff, others not so much so.
what about like stimulants like Ritalin? What does that do to them?
doesn't that affect the synaptic gaps?
I don't know what relevant effects, to be honest. I know why it's prescribed, why it's overprescribed, why people take it, but I don't know if it enhances any type of brain function. I know it's supposed to quell or kind of quiet certain types of brain function. Synapse, gap or space between neurons. We always used to joke in graduate school, kind of tailing what you were just asking. The students who weren't quite getting it, problem was they just had wide synaptic gaps.
Yeah.
It wasn't making it. Two different types. There is an electrical synapse and a chemical synapse. Electrical synapse we really don't talk about much. They're rare, occurring in animals which practice rapid movements. Sometimes the wings of insects beating, or a hummingbird, beats so fast, it beats faster than the signals can actually go from the wings to the brain and back. So it's more on some type of synapse that actually works very, very quickly. And electrical synapses tend to work more quickly than chemical. Most of the reactions in our body don't require that kind of rapid rate. And so that's why we usually use chemical synapses. That's what we're going to be talking about. Try that again. The chemical synapse, a neurotransmitter is released from the presynaptic neuron. Neurotransmitters package chemical in vesicles at the edge of the synaptic gap. So, here is my neuron up here, let's pretend. Here is the axon coming down. Here is the into the axon, the synapse, and here is the let's just pretend this is another neuron, the postsynaptic neuron. And so what happens? The signal comes down, but it stops right here. The signal can't jump itself from the pre to postsynaptic neuron. But what it does is it causes these vesicles containing chemicals to move towards the end of the neuron to release the neurotransmitter that carries the signal from the pre to the post, binding to receptors, and then the signal continues on down. So the neurotransmitter continues the signal from a neuron to a neuron or a neuron to an effector cell. So you've got this gap. The signal comes down, reaches the gap, the neurotransmitter is released, it bridges the gap, and the signal continues on. Like I said, to the next neuron or to the effector cell. It's muscle, endocrine, whatever. There are lots of different types of neurotransmitters. I'm going to talk about 3. First up is acetylcholine. Excites skeletal muscles and reduces the strength and rate of cardiac muscle contraction. You find acetylcholine in the neuromuscular junction. It could be skeletal muscle, it could be smooth muscle, it could be cardiac muscle. Acetylcholine is one of the more common neurotransmitters. Dopamine, serotonin affect sleep, mood, attention, and learning. You find this mostly in the brain. Endorphins decreases pain perception while producing a sense of euphoria. Runner's high. Anytime you work out, exercise, push yourself hard, you get to a peak, a plateau, where all of a sudden you feel like you've broken through some kind of barrier, and you start to feel pretty good. It's like, yeah, I settled in. Now I've achieved my exercise goal or whatever it is. But what it is endorphins being released, kind of masking the pain and giving you a sense of euphoria, a sense of feeling good. Not necessarily a bad thing. So, the signal comes down, it's like I showed you before. It pushes these vesicles to the membrane surface of the presynaptic axon. It releases the neurotransmitter across. The dendrite on the other side has receptors on it, picks up these neurotransmitter molecules, and the signal continues. Now, give me something to talk about. I'm surprised because heroin's getting really big in Northern Kentucky, Greater Cincinnati right now. Seriously, every day in the newspaper there's an article about that.
Like if someone's on drugs and like they don't really feel anything, is it because, you know, like you,
Because you've got the wrong neurotransmitter, the wrong chemical in the receptor site, so it's not bringing the correct signal to the brain.
so like you try to take someone down that's on drugs, but they're not feeling all the blows because they're preoccupied. I see.
Exactly. So anyway, that's how those work. Okay, now we're going to shift into more of the the anatomical side of the vertebrate nervous system. First up is the central nervous system. This contains the brain and the spinal cord. Located on the dorsal side of the body. Where's the dorsal side? Front or back? Back. And you're probably thinking, where else would it be? Some organisms have the nervous system on the ventral side of the body, the belly. Front. Ours is on the back. Surrounded by cerebrospinal fluid, CSF, a blood filtrate. Why don't you want blood in or around the brain and the spinal cord?
No immune system.
What?
You don't want the immune system in
You
your.
don't want the immune system there. What did you say, Juan? They carry some pathogens into the brain. The other big thing is, let's say you get a brain injury. And one of the other functions of the immune system, white blood cells, is to remove damaged cells. So let's say you get a little brain injury here, and the white blood cells start eating away at those neurons. And they actually do more damage than good. Because your brain has the ability to sometimes rewire itself, working around the damaged cells. But you eat away too many of the damaged cells into the good cells, and then the rewiring doesn't work, and it actually makes the situation worse. So you want to keep the blood out of the brain and spinal fluid. So the cerebral spinal fluid, plus it actually kind of floats the brain in your skull, making your brain a little bit lighter. The peripheral nervous system consists of cranial and spinal nerves. You've got fingers and toes, I know, really, that have nerves running all the way to them. Okay? Your central nervous system stops the brain and the spinal cord. So how do you get to your fingers and toes? Well, that's the job of the peripheral nervous system. It gets us out there and beyond. Cranial nerves originate in the brain and terminate in organs of the head and upper body. So what they're talking about is you have nerves coming off your brain. And they go to things like your eyes, your ears, your nose, your tongue. And that's okay, fine, I get that. Pretty much all the muscles in your face that control look, to smile, to frown. All this stuff and some of them extend down into parts of the neck. And then you have the spinal nerves that originate in the spinal cord and terminate in the lower body. You have concentrations of spinal nerves in and around the shoulders and the hips because you have a lot of information coming into those regions from your fingers, from your toes, from your arms, your legs. So you have concentrations of spinal nerves in and around the, like I said, shoulders and the hips. So the peripheral nervous system is made up of the cranial and spinal nerves. So you got the central nervous system and the peripheral nervous system. And you're thinking, so far so good, I got this, I can handle it. The peripheral nervous system is going to be divided into two systems. And then one of those systems is going to be divided into two more systems. And I will want you to know all the different kinds. They're actually pretty cool. Yeah.
If you don't allow blood to the spinal cord, how the biggest infection, how does your body react to the infection?
Well,
Meningitis.
it can't really react to it because it can't get there. You basically have to treat with, depending on the type of infection, antibiotics or drugs. Like, let's say you develop meningitis, which is an infection of not so much from the brain, but the meninges, the tissue surrounding the brain. And you've got to treat that with a lot of serious drugs to try and clear that up because it can be lethal. So yeah, it's just, your body really can't deal with that. It has to be some kind of external introduction of drugs. Okay, first up, somatic and automatic nervous systems. The somatic nervous system carries signals to and from skeletal muscles, mainly in response to external stimuli. This is under voluntary control. Somatic, soma is Greek for body. This goes to the body, the skeletal muscles in particular. What you have is basically a neuron with a very long axon that terminates in the muscles. And that's going to be significant, so just hang on to that for a second. It's going to be very heavily myelinated because you need precise movements in some of your skeletal muscles. And so you want to make sure you have a very clear and distinct signal. This ACH is the neurotransmitter acetylcholine. You don't need to know that. You don't need to know everything I'm telling you. I'm just kind of filling in the blanks for you right now. We talked about some of the axons were very heavily myelinated, some not so much. But to ensure that you get a good signal, no disruption, you get one long axon, heavily insulated, heavily myelinated. The other half is the autonomic nervous system. Sometimes I like to call it the automatic nervous system because this is involuntary. Regulates internal stimuli by controlling smooth and cardiac muscles and organs of digestive, excretory, endocrine, and cardiovascular systems. Okay, I'm pretty sure that you're good with digestive, excretory. I keep talking about endocrine. What's the endocrine system? Hormones. So everybody's on the same page now. That's the hormones. And the cardiovascular system. This is involuntary. Notice, very light myelin sheathing on all of these. Notice also that sometimes you go from one neuron to another before you terminate where you're supposed to go. Light myelin sheathing, here's the glands that secrete whatever you need. Light myelin sheathing from one neuron to another neuron before you reach things like the heart, other organs. By the way, is norepinephrine. It's the opposite of epinephrine. And then ACH again for acetylcholine. Why would these seemingly important neurons be lightly myelinated?
Because they're involuntary.
Okay,
No?
no, that's true. And so carry it a step farther.
So they don't need the full signal to know exactly what to do. It's just kind of already happening. It's already.
Okay, it's very repetitive.
Mm.
If you do the same thing over and over and over again, you don't really need to pay attention every time you do it. I mean, maybe subconsciously you do, but you don't have to all the time. It doesn't take that precise movement every single time. The nice thing about this is, because it is involuntary, you don't have to think about digesting food. You don't have to sit there and think about making your heart beat lub-dub, lub-dub, lub-dub. Okay? It's all under involuntary control. And because you digest food on a regular basis, because your heart beats every second of every day, The body knows what it needs to do. It's not going to change up too much. That's why, like myelin sheathing, and that's why you might have breaks in the neuron going from one axon to the dendrites of another, instead of going directly to like you do with the muscles. The autonomic nervous system can be divided into two parts, the sympathetic and parasympathetic. Sympathetic division corresponds to arousal and energy generation, also homeostasis. Walking down the street at night, you hear a dog bark. You don't know if it's tied up, if it's out, if it's a nice dog, if it's a mean dog, you freeze in your tracks. But you're not just standing still. All of a sudden, you have epinephrine, formerly known as adrenaline, coursing through your blood. Because it's a fight or flight response. Do I try and run away? Do I stand my ground? Do I take the dog? Don't know. Your brain becomes more alert because it's releasing sugar into your brain. Your digestive system shuts down because you're not going to be digesting food right now. Your muscles become a little bit tense. You have more sugar going to them. You're prepared to do something. That's the sympathetic division, causing all this to happen. Parasympathetic division corresponds to calming, returning to normal functions. You just eat a big Thanksgiving meal, and you don't feel like doing anything. Putting on a really mediocre football game and falling asleep to it. That's parasympathetic. And I keep saying, also homeostasis. What's homeostasis? Same steady state. Maintaining an even strain. So if you get too low, the sympathetic division brings you back up. Or if you get too high, the parasympathetic division brings you back down. That's what it means about homeostasis, keeping you around basic parameters. So we've got the central peripheral nervous system. The peripheral nervous system can be divided into the somatic and the autonomic. The autonomic is broken down into sympathetic and parasympathetic. Make sure you can distinguish between each of these. They're in pairs. Learn them that way. Diseases and disorders. First up, bipolar disorder. I'm going to give you some basics about it. Some of you might have experience with this. I'm not talking about personally. You don't divulge any personal stories about this. It's general ideas, causes, treatments. It may not be the most current. It's what's been available, say, in the last 5 years. Relatively current based on what the textbook says. First up, bipolar disorder. Mood swings, high self-esteem, increased energy, flow of ideas, increased creativity, increased risk-taking, constant conversation, promiscuity, reckless spending. These are all examples of bipolar disorder. Mood swings. You're up one minute, you're down the next. Cause unknown. Okay, there could be a number of different causes. One of the treatment is Prozac. Lithium is another treatment for this stuff. What's the downside to Prozac?
Side effects?
Yeah, such as?
I don't know, bleeding of the eyes.
Yeah, don't smell that. Okay, any kind of treatment brings this down. And some of the more creative people
They get depressed.
have bipolar. Do what?
Would they get depressed?
They could be depressed. Because what you're doing now is bringing them down, trying to keep their enthusiasm, excitement, getting out of control. And that also affects the flow of ideas and increased creativity. Let's say you're a writer or an actor or a musician, artist, and you go on this stuff because you have bipolar disorder. And now all of a sudden you can't write, you can't act, you can't make music, you can't draw or sculpt or do anything like that. And so it's adversely affecting your life. So you come off of it to do this stuff, and then you have all the problems. Yes. Right. The mood swings go the other way and go down. You can bring them up. I'm not saying don't take it. Okay. I think it's the best thing you've got going right now. But for some people, that's why people who are really good in their fields, or maybe what we consider a little eccentric, and we just think of them, oh, that's just being artists, but it could be at least maybe minor bipolar disorder that makes them so creative, that makes them so, risk-taking.
Kurt Cobain.
And all that. allow them to do all that they do.
Naming people that we think.
Schizophrenia. Unable to distinguish reality, hallucinations, voices, delusions, paranoia, blunted emotions, easily distracted, lack of initiative, and lack of conversation. Again, cause unknown. Treatment, dopamine receptor blockers. So unable to distinguish reality, they tend to hallucinate, hearing voices, they have delusions, which makes them paranoid, blunted emotions, easily distracted, lack of initiative, and lack of conversation. Yes.
I just want to know, what could it be if somebody says that they see like people's auras, they see like flushes of color just on a day-to-day basis, or
Say that again. They see what?
like they see like outlines of people's auras, like they see like
I used to work with somebody like that. I don't think that's so much schizophrenia.
No.
I think that's just a perception. It could be a visual thing. It could be a misperception of visual stimuli. So, I don't know.
Are people able to take, like, say if they were like drug addicts or something like that, would they be able to take enough drugs to become schizophrenic or bipolar?
Sure, you can damage
Yeah,
receptors if you abuse drugs.
Okay.
I think once you add drugs into the mix, it kind of alters,
What type of drugs would it have to be though? Like
your
just
pot can do that even if you smoke a blunt.
Yeah, within the psychology.
They
I
have
think
to
we're not
Somebody's on
legalizing
drugs,
that
yeah.
anymore in the U.S.
Can't really diagnose
One of the
it.
things about these drugs, you talk about addiction and everything like that, things like Prozac and some of this other stuff that people take, once people get on the stuff, they start feeling good, and then they decide they don't need to take the drugs anymore because the drugs do have side effects to them, and so they come off the drugs and they go through the whole process again. That's why sometimes you might read about people who may be homeless and they were under the care, they were taking their medications, and all of a sudden they lost contact with family, with friends. They go off the drugs, they go homeless again, and they have all kinds of issues, problems, arrests, things like that. It's a difficult situation. It's difficult to treat these things and to maintain a person's stability. Yes.
Can this be found in other mammals, or is it only specifically to humans?
I don't know if enough research has been done in that. I mean, certainly, let's take pit bulls, for instance. There are some pit bulls that are very docile, some that are very aggressive. Could it be that they might be suffering from schizophrenia or bipolar disorder of some kind? Perhaps.
To make Prozac for dogs.
Do they really? Puppy Prozac.
Chicken flavored.
Chicken. What was that?
So dopamine affects sleep, mood, attention, and learning. So dopamine receptor blockers, obviously they're getting too many stimuli in here, and we want to try and block that somehow, some way, so the dopamine doesn't adversely affect them. Dopamine's going to come up again in a second right here. Parkinson's disease, progressive motor disorder characterized by slowness and difficulty in initiating movement, rigidity, masked facial expression, muscle tremors, poor balance, fixed posture, and shuffling gait. It doesn't really affect the mind so much as it does the body. And it might just start off as somebody might have a hand that shakes or a head that kind of shakes back and forth a little bit. But then it's progressive. It advances. It gets worse. Neuronal death, loss of dopamine receptors. So treatment, dopamine agonists. Are you pro or con if you're an agonist? That's an antagonist. Agonist before. So what you're trying to do is introduce more dopamine to compensate for the loss of receptors, kind of flooding the system with dopamine, trying to fix this, but it doesn't always work. Muhammad Ali, Michael J. Fox, two of the more famous people now who have Parkinson's. Of course, Michael J. Fox has the Fox Foundation trying to find a cure for it. Yeah.
I heard that use of like cocaine or drugs that increase dopamine can cause Parkinson's disease.
It could. I mean, neuronal death, loss of dopamine receptors. Most of the time, though, they're not sure what causes it. There could be a genetic component to it. It could be that it's just one of those things. But this and some of the other stuff, the age-related diseases, because our population is getting older, you're going to see a lot more research going into things like Parkinson's and Alzheimer's, trying to figure out how to resolve these situations.
I was watching NPR the other day, and they had like this whole thing on Alzheimer's where they said it was caused by a virus, and it's actually like a catchable disease that they don't think it's genetic anymore.
That's news to me. I've not heard that.
It was the strangest thing. They said that like that's why it's becoming more and more common.
Well, I've heard that it could be what you eat or don't eat. It could be head injuries that could cause it. That's why they're really looking at football players, former NFL players, and when one dies, usually in their 50s or 60s, well before the normal age of males in the U.S., they try to take their brains and look at them. One of the biggest studies was a bunch of nuns who donated their brains to science and research of Alzheimer's. Not that they have a lot of head injuries that I know of. Yeah.
So the biggest difference between Parkinson's and Alzheimer's is the location effects?
Not so much. This is the neuronal death. With Alzheimer's, you have plaques in the brain start eating away at the brain, which starts to be holes in the brain. We're going to talk about that next. Progressive mental deterioration, maybe sometimes it was formerly known as dementia or senility, before we really knew what it was. Characterized by confusion, memory loss. Usually the memory loss is from front to back. They might remember things that happened, years ago, but they can't remember what they had for breakfast. Inability to recognize people. Sometimes personality changes. Some Alzheimer's patients become aggressive, even when they were never aggressive in their life. Physical incapacitation and deterioration. Both of these ultimately, Alzheimer's ultimately affects the body. Even though the mind starts to go, eventually the body deteriorates. With Parkinson's, as the body deteriorates, the mind slowly starts to go. So ultimately, they both affect the entire person. Neuronal death, again, brain shrinkage, treatment, Aricept in the early stages, nothing to date in the late stages. So if you catch somebody early on Alzheimer's, Aricept might keep them a little more focused, keep them of who they are, but ultimately, as Reagan called it, it's the long goodbye. You're just going to slide into nothingness.
There's a medical term for it, starts with an A, I believe. It's when the brain starts shrinking in the front of the frontal cortex.
I'm not sure. The only thing that's popping in mind is anencephaly, but I'm not sure if that's it.
Anencephaly?
Anencephaly.
Do their brains kind of shrink a little bit when this happens?
It ends up where they actually have plaques. That's what they call it. When you have a plaque, it's like they have a hole in something. And it's starting to cause the brain to deteriorate. But like I said, this with Parkinson's usually affects the older population. Right now, the older population is bigger than it's ever been. So you're probably going to see a lot more research going into these areas, trying to resolve. Yeah.
Yeah, but that's not just natural decay of the body after a while? No? Okay.
It's more rapid decay.
More rapid decay?
More complete decay, too.
Okay.
In fact, you have people who die like completely mentally shocked, even though they may be very, very old.
Mm-hmm.
In this case,
No, not with Alzheimer's, just like normal population. You have people who die that don't lose their mental function. It's die old age.
no, yeah, some people can remain sharp to the end. I've got a friend of the family back home who's 102, and she's still sharp as can be. Her body's not keeping up with her, but she's still pretty sharp.
Well, isn't death of old age that's something going on with your body? You're not just passing away.
It
Something.
depends on the individual, but these diseases accelerate that rate of decay.
Okay.
Okay, transitioning. We've got done talking about the technical stuff. Now we're talking about what uses the technical stuff has. Mainly to begin with sensory receptors. Sensory transduction, conversion of stimulus energy into change in memory potential of a sensory receptor. Kind of tying the two together now. Okay, so we're working off the whole idea that everything uses action potentials to get the stimulus to the brain. But different parts of the body, different senses, uses these action potentials differently based on the stimulus. Amplification, strengthening of stimulus energy by cells and sensory pathways can increase from tens to hundreds of thousands of times. Different animals exploit different senses better than others. We don't have a great sense of smell, but dogs have a really good sense of smell. Our sense of vision is not the best, but it is in color. That's why our sense of smell isn't so bad. A long time ago in our evolutionary history, we decided to follow color vision rather than our sense of smell. We decided to be more active during the day than at night, and that's what helped sculpt that sense. Different animals rely on different senses. And the more you're dependent upon something, the better developed it should be through evolution.
So theoretically, we could change like 100,000 years if the environment changed. It was more darkness and we would develop a sense of smell.
We could do that, or we could become more adapted to seeing at night. I'll tell you how that happens. So sensory transduction, conversion of stimulus energy into change in membrane potential of sensory receptor, extremely sensitive. Amplification, strengthening of stimulus energy by cells and sensory pathways. We'll talk about all this. So, sensory receptors, transmission, integration, sensory adaptation. Transmission is the conduction of sensory signals to the central nervous system by neurons or neurotransmitters. Nothing too big there. So we get the signal from the outside, we take it inside, and our brain processes it. Integration is interpretation of sensory signals within processing centers of the central nervous system. Sensory adaptation. Increase in responsiveness during continued stimulation. Can you guess what the one sense is that can actually adapt to a stimulus over time?
Smell.
The nose.
Smell.
That's why it's got clothespin on its nose. Because it adapts. It's the irony of the picture. Eventually, over time, you will stop smelling things because your nose adapts to it. We talked about this in here before, how some people put on way too much perfume or cologne because they don't smell it anymore. They don't think they have enough on. You find this especially in older people. They just don't smell it anymore because their nose becomes desensitized. It is the only sense organ that adapts. If you have a loud noise blaring in your ears, you're still going to hear the loud noise. If you have a bright light flashing in your eyes, you're still going to see the bright light. If somebody's sitting there with a hammer on your hand, you're still going to feel that until they break everything in the hand.
What if you don't really have a strong sense of smell, like at
Some
all?
people don't. Some people have a better sense of smell. I mean, you hear about people who might work for a fragrance company or a good sense of taste might work for a winery or something like that because their senses are extra perceptive in those areas. But,
Because me, my dad, and my brother, we have the worst sense of smell.
okay, not a bad thing.
And like my dad can't smell at all.
Your dad what?
He can't smell at all.
Okay, it happens
Yeah,
sometimes.
I don't know how that happens.
Either it didn't develop or something happened to damage it over time.
Mm-hmm.
I've got a bad sense of smell, but I've learned my allergies on that because I think, having so many allergic reactions and related problems when I was a kid, it just kind of deadened everything. So like I said, the nose is one of the few that actually adapt, becomes desensitized. Everything else still stays pretty much alert. Okay, two different types of sensory receptors. We have chemoreceptors and mechanoreceptors. Kind of makes sense in the name. Chemoreceptors transmit information about total solute, concentration, or individual kinds of molecules. That depends on how good your sense of smell is. You walk into a house, let's say somebody's cooking soup on the stove. You walk in, vegetable soup. Dog walks into the house and vegetable soup. It's got tomato and beans, and it's got corn, and it's got beef. All right, score. In other words, you smell the soup, the dog can break down the individual ingredients of the soup. Their noses are that much more sensitive. Mechanoreceptors sense physical deformation caused by stimuli, pressure, touch, stretch, motion, and sound. Pressure. Okay. Whiskers of a cat or a dog, for that matter. They're here like an extra pair of hands on the side of their face. They can feel things out there. Touch. Anytime you touch something, you touch a wall, and you can feel this is a very solid thing. You touch your skin, not quite so solid. Okay? That touch. Stretch. Anytime you stretch your muscles, you can feel that stretch. Again, it gives your body a sense of orientation, where you are in time and space. Motion. The formation of the eye as it moves to adjust to something. I don't mean just the eyes moving back and forth in your head, but as the lenses focus in your eye. And sound. You've got the tympanic membrane, and as the sound waves hit it, they cause it to vibrate. And there's 3 little bones on the inside of the tympanic membrane that sends those vibrations to the brain and the signal the brain can perceive as sound. So all of these are examples of mechanoreceptors. 2 chemoreceptors we've got: sense of smell and sense of taste. Another area of receptors are electromagnetic receptors. Now, we don't have all of these, but we do have some of them. Detect forms of energy, including visible light, electricity, and magnetism. Photoreceptors we're pretty good at with light. Infrared receptors, yeah, we're not so good at that. We can't look at somebody and see their body heat, although maybe people detecting auras, that's what they see coming on. Who knows? Electroreceptors are electrical impulses. Talked about that with sharks. They can detect weak electrical stimuli in the water from these electrical impulses. We can't. Magnetoreceptors, some animals can tap into the magnetic fields of the Earth. They know where the magnetic North Pole is. Pigeons, they believe, are pretty good at doing that. A lot of migratory animals are pretty good at doing that. Not so much us. So electromagnetic receptors detect forms of energy, including visible light, electricity, and magnetism. Bless you. Okay. Sensory receptors in the skin, we've got thermoreceptors and nociceptors. Thermoreceptors, by the name, detect how hot or how cold something is. Nociceptors detect pain. Nobody put these eyes on here today. Surface and body temperature. So you've got these on the surface of the skin to detect what the temperature is like above the skin, and you've got them deep in the body. Now that will help to maintain your body temperature. Nociceptors are pain receptors in a class of naked dendrites in the epidermis of the skin. When they talk about naked dendrites, we're saying that these don't have a lot of sheathing around them. They've got hardly anything, making them raw, exposed nerve endings. So here's a piece of skin. Hopefully it looks familiar. We did in lab 2 weeks ago, right? You'll be on the quiz this week, maybe. What? You don't remember?
No, I was kidding.
What'd you say?
I don't remember, but I'm sure.
Were you really?
Yeah.
Okay. That was a rhetorical question. I don't remember the answer. Okay. Here on the surface of the skin, you've got these naked dendrites here right below the surface. And they're very sensitive to pain. Now, deeper in the skin, you've got cold receptors and heat receptors. Okay? So the nociceptors are right here, the hot and cold receptors down there.
Where's the sweat gland?
Seriously?
That's what I thought.
What kind of gland is that?
Back gland?
A yellow one.
Yo.
Wait for the future.
I can indirectly answer.
Okay, we'll do these 2 things and we'll quit. Not these two things, the next slide too.
Oh.
Seriously? Because we're going to quit, right? And you're disappointed about us stopping, right?
Yeah. We don't want to stop.
Okay, how do you know what's up and down? How do you know what's left and right? How do you know if you're standing upright or bent over? Well, different organisms detect this in using different things, but the way is basically similar. And invertebrates, animals without backbones, they have statocysts. Sensory organs contain mechanoreceptors that detect changes in gravity. Okay, this chamber here is a statocyst. All right? All these cells have tiny little nerves in here. And if you talk about, take a ball and make it up of a lot of tiny little cells. And each cell has these nerve endings going inside this ball. And you fill the ball with something like BBs. And as you move that ball around, those BBs are going to move around. And as they move around, they're going to brush up against these nerve endings. And it's going to tell the ball, that chamber, where the BBs are in relation to where they should be. So if you take it and leave it upright like this, they should be on the bottom. That's normal. If you turn it sideways, the BBs start to slide sideways, and that's going to send those nerve endings to the brain saying, hey, you're on your side. Turn it all the way around upside down, then it's going to say, hey, you're upside down. Stateless is a disc granule for the BBs in a chamber that detects gravity by position. And you see this incrustations. So this is the statolith in here. And as it rolls around, as he changes position, then it's going to tell him what position you're in. That's how that works. That's in invertebrates. In vertebrates, we have something similar, but because we're vertebrates, it can be more complicated. Uricule, or the saccule, is the same thing as the statocyst. It's the chamber in the inner ear that responds to changes in head position with respect to gravity. Inside this, instead of statoliths, we have otoliths. Otto refers to the ear. Calcium carbonate granules in a chamber that detect gravity by position. That's in humans. So you've got these little crystals in here. And as you move your head sideways, they're going to slide sideways. As you move it back up, they're going to slide back down. They're going to tell you you're upright. You move to the other side, they're going to do the same thing. They just have a blast when you ride roller coasters that have loops in them. Okay, they're just bouncing around all over inside these chambers. That's why when you get off, you might not really have your bearings at first. So these two things, in invertebrates, you have statocysts and statoliths. It's a basic design. It's basically the same design as this. It's just a little more complex. You have the uricule and the saccule, which is the chamber, and then you have the otoliths inside. But you can see how each cell has a nerve ending that comes up into it, detecting where these are inside the chamber, just like the other did. Oh, good place to stop. We'll pick up with auditory receptors after the quiz on Thursday. Talking about auditory receptors, we're basically talking about things with which to hear. Hairs of varying length for varying frequencies, as well as a tympanic membrane covering an internal air chamber. What we're talking about in this case is the house cricket. Here is the tympanic membrane. This is actually located on one of its legs, and that's where it hears. Like I said, it's going to be different for different organisms. Ours, obviously, aren't on our legs. They're with most of the rest of our sensory organs on our head. We became cephalized. Not civilized, cephalized. So we still haven't achieved that yet. Hairs of varying length for varying frequencies, as well as a tympanic membrane covering an internal air chamber. What happens is this membrane vibrates. It sends signals by one means or another. We'll talk about a couple of different means how that occurs. Through the nervous system to the brain, so the brain can perceive the sound waves and make sense of it. Vibrations perceived by tympanic membrane are transmitted across bones in the middle ear. You don't need to know these, but if you ever want to, they are the stapes, the incus, and the malleus to the inner ear. Hearing ranges, humans at about 20,000 hertz, canines at about 40,000 hertz, bats at about 100,000 hertz. We all know dogs can hear better than us. Hearing's about twice as good. Still not the dog's best feature. They have something else they rely on more. Bats mostly hearing, all about hearing. They communicate in that echolocation, which is in a range most of us cannot hear. It's well above our range of hearing, but it works for them very nicely. They come out at night. You ever see the eyes of a bat? They're very small, very insignificant. They rely on echolocation to kind of paint their world for so they can see what's out there, so they can use it to hunt. And yes, I'm a big enough geek that I've got what they call a bat detector. No, it has nothing to do with Batman. I like I'm running around in costumes at night. Basically what it does, it looks like an old transistor radio. You might have seen some of these, maybe you have. And what it does is it tunes into the frequency of the bats. And you can actually hear them hunting. And it sounds like a series of clicks. And when they get closer to their prey, let's say a moth or a mosquito, the clicks get closer together. It almost sounds like a zipper at the very end. And then it goes silent because they've caught their prey. And then it'll pick up again. Sometimes you'll hear the bats communicating with each other. You'll hear 2 or 3 at the same time. It's actually pretty cool. Based on the frequency, you can actually tell what species of bat it is, which is actually also kind of cool about this. So what happens with us, the sound comes in the auditory canal, it vibrates right here against the tympanic membrane. Here are the little bones right here, tympanic membrane, the 3 little bones right here. As they vibrate, they send signals to the brain through a nerve, and that's how the brain takes what you hear and changes it into something that actually makes sense to you. Now, when we talked about the parts of the ear where we have that which gives us orientation, the auricle, the saccule, that's in this region right around in here. Yeah.
Say it's like really silent, and that bat's like, does it need to make noise so that it can hear so it can see where it's going?
Sometimes. If it's in a big open space, it's not going to make as much noise, but if it's in
Just
places
probably
with
the
more
flapping.
obstacles, it's going to make more noise.
The flapping of the wings might make echoes off of things.
So it listens for the
Not
echoes?
so much the flapping of the wings, it's just that echolocation. And it's a high-pitched sound, and it bounces off things, and it comes back. If you look at bats, most of them have very large ears, and sometimes their faces are even formed to take the sound and funnel it into their ears, so they can perceive the sound better. So it's all about sound with them. Emitting sound and hearing the sound come back. Now, air, swim bladder, and the Weberian apparatus. We talked about the air, the swim bladder when we dissected the fish. And it uses its swim bladder not only for buoyancy, but it also serves like an eardrum, basically. Vibrates in response to sound, transfer to inner ear. Kind of funny to think of fish with an inner ear because they don't have these outer ears. In fact, mammals are the only ones that do. Think about it. The Weberian apparatus conducts vibrations from the bladder to the inner ear. Ossicles, which refer to tiny little bones. And so I have a series of tiny little bones. This is the vertebrae. The Weberian ossicles are these tiny little bones on each side here, not unlike our 3 little bones that vibrate in relation to the eardrum. So the swim bladder picks up vibrations. In a second, I'll tell you how the swim bladder gets the vibrations. But it vibrates, and it causes these bones to vibrate, sending the signal to the brain so it can figure out what kind of noise it's making. Here's part of the inner ear. You've got the semicircular duct, the sacculus. Go back a second. Semicircular canals. We talked about the auricle, the saccule in us. So we're seeing a lot of similarities. Even though it's something that lives in an entirely different environment, evolutionary chain is much earlier than we are, been around for hundreds of millions of years, they still use basically the same system with which to perceive sound. The big difference is they pick up vibrations in water, we pick up vibrations in air. So everything for them is geared towards an aquatic environment, even the way they pick up sound. The lateral line system. This is what connects the bladder to the external environment. Along the side of most fish, you see a very faint line. Sometimes it's almost imperceptible, other times it's very obvious. Maybe below that line it's very light, above the line it's very dark. But this lateral line system, and you you see it down here. Here's the pores leading to the outside, these external openings. Water displaces. Well, what causes water to displace? Well, if you get in water, you stand real still and the fish swims up to you, you go to try and grab it or something like that, it moves because even before you put your hands in the water to grab it, it can detect any vibrations coming off your muscles in your legs that might tense up. And that's how they pick up the sound too. So it enters through these holes in the side. It's like tiny little ears all along the side of the fish, along this lateral line. It goes through here, and it starts to trigger a signal here on the neuromast. And this is what it looks like, this little projection into this canal here that picks up the vibrations. They have sensory hairs here that vibrate, and then it sends it to the nerve. The lateral line system. Similar to the inner ear. What?
Are fish the only ones who have this?
Sharks have it too. Those kinds of aquatic animals. Dolphins do not. That's what you're thinking. So it's not all aquatic animals. Fish and sharks, yes. Okay, changing gears. Gustation. Ability to taste chemicals present in solution. We have modified epithelial cells located on papillae and organized into taste buds. The papillae are on our tongue. 5, bitter, salty, sour, sweet, umami, which is for delicious, and possibly a 6th, savory. Savory. So, in case you didn't know where your tongue was, this diagram shows you right in your mouth. You have these taste pores. You have molecules that trigger your taste buds. You have sensory receptor cells. You have sensory neurons that go from these taste buds to your brain. And it tells you what you're tasting. Are you starting to see a pattern here with hearing, with taste, in a few seconds with smell, where you have some kind of structure that perceives the stimulus. And then you have nerves that go to the structure, and they lead from the structure to bigger nerves that eventually go to the brain. So the processes are very similar as to how we perceive stimuli in our environment, whether it's sound, whether it's taste, whether it's the way we smell. It all works about the same. Yeah.
Do you know, are humans, do they have like the broadest range for like, taste?
I don't know. To be honest, I've never known if that's been tested. I'm sure it has. But to what degree and what other animals, I don't know. I know, like with birds, probably not a lot of taste there. Because let's say you've got a bird feeder with the rats, the ground squirrels, something's getting into it. You put like cayenne pepper or some kind of hot spice of pepper around it. The mammals will leave it alone, but it doesn't affect the birds. So I'm guessing mammals are probably a little bit better than most species. But who's the best in the mammals? Is it humans? Is it the primates? Is it dogs, cats? I don't know. Good question. I hope you have an answer. Olfaction. Ability to smell airborne chemicals. So taste in solution, olfaction, sense of smell in air. Neurons line the nasal cavity, sending impulses directly to the olfactory bulb in the brain. We might see this, but definitely see the optic lobes when we dissect the brain next week in lab. Oh, by the way, I've posted something that might help you with lab for tomorrow and next week. Tomorrow we're going to start dissecting the fetal pigs. Yes, they came in. And we're also going to look at the sheep heart in conjunction with our dissections tomorrow. The following week, we'll be looking maybe again at the pigs, but at the kidney, the eye, and the brain. And I've got all that posted on there for you. I don't have the kidney, but I think the lead name covers it pretty well. Anyway, back to this. Neurons line the nasal cavity, sending impulses directly to the olfactory bulb in the brain. Okay, so things come in odorant. Comes in through the nose, the nostril, into the nasal cavity. You've got bones lining the nasal cavity, and extending down between these bones you have epithelial cells and the chemoreceptors extending beyond those. So they actually extend into this opening here. Mucus. It helps if your nasal passages are moist in order to better perceive this. If they start to dry out, they may not work as effectively. just like before, you've got tiny little nerves going to bigger nerves, eventually going to the olfactory nerve, which goes to that part of the brain that determines what it is you're smelling. Now, some people have better sense of smell than others. I think we might have touched on this, maybe not. Where you might have some people who are hired by fragrance companies to smell the quality of fragrance, by wine companies to taste the quality of different wines because they've got extra perceptive senses of smell and taste. Some people have lousy senses of smell and taste. Now, 30% of human genes deal with olfaction, distinguishing thousands of different odors. Taste and smell, though independent, interact. So, what that is saying, this last statement, kind of important. You don't need to know it for an exam, it's in italics, but it brings home a point. You don't need to smell and taste things for both your nose and your tongue to work properly. But, have you ever noticed, if you have a really bad cold, your head stopped up, food just doesn't taste like anything, it's just really bland? That's because over time we've become independent or interdependent on smelling what we taste. I mean, sometimes you smell something that's cooking and your mouth might start to water. Okay, you're starting to perceive the smell before you taste it, and then you eat it, and it can start to water all over. Okay, so even though they are two distinct parts of our sensory system, oftentimes they work together.
It sounds like so we can smell. So we can taste what we smell and smell what we taste.
Kind of, sort of, yes. And that's why if you have a cold, if your nose gets stopped up, your sense of smell isn't working too good, sometimes you can't taste it as well either, just because we've become dependent upon both of those working together. Just something we've trained our brains to do. Something like that, yeah. Hold your nose and eat something you don't like, maybe it won't taste bad.
Or medicine.
Okay, vision. We're going to start with invertebrates and work our way up to invertebrates. The ocelli. Light detecting organ that determines light intensity and direction. It doesn't have any image forming capabilities. It's just a light sensor, a photosensor. The ocelli between the compound eyes of a damselfly. Here is a blue ocelli, another one, and a black one. Ocellus. So this one has 3. They don't always come in 3s. They might be more, they might be fewer. Different colors. This one just blends in with the head of the damselfly. So 2 blue, 1 black ocelli. Only detects light. You find this in insects. We don't have any ocelli in us. We're just dealing with invertebrates. Also, same picture, compound eyes. Thousands of light detectors, each with its own light focusing lens. What it does, and you can kind of see in this is why I left it up here for, shows you the compound eye. And you see that it's a mosaic. It's like tiny little tiles all put together to make each big eye. Mosaic image, so each little lens presents an image to the insect, but its brain takes those tiny little images and makes one sharp image out of it. Capable of detecting motion, color, and ultraviolet light. Motion and color we can pick up on, no problem. Ultraviolet light, yeah, we can't see that. But there are actually some plants that put out ultraviolet colors or ultraviolet radiation that we cannot pick up on. But certain insects can. It gives them an entirely different perspective of the plant. They view the plant in entirely different terms. Some birds can detect ultraviolet light, and some bats can as well. Usually these are pollinators, by the way. Yeah.
What are the benefits of being able to detect the ultraviolet light?
Like I said, some plants put out colors in ultraviolet light, and the insects can detect them. So it's just another way of the plant and the pollinator getting together. Yeah.
So within each one of those little dots right there, you said there's thousands?
Hundreds, maybe thousands of
Okay.
And each one gives the insect an image, but what it does is its brain takes that image and puts them all together into one cohesive picture. Think about like when you have a satellite taking pictures of, say, Mars. It's not going to take one big detailed picture, it's going to take a lot of little pictures. And then computers take that and put it together into one cohesive image. Well, that's basically how this works. Each little lens captures an image, and chances are it's going to be almost all the same image, just repeated 100 or 1,000 times.
Okay.
And then what happens is the brain takes that and puts it into a form that the insect can perceive. Whether it's looking for a plant, looking for prey, avoiding a predator, whatever the case might be, it allows it to see that.
Okay.
And again, compound eyes, just like Ocelli, are found in insects. Yes.
Are the brains of the insects with the compound eyes, like, do they interpret the images faster, or?
I don't know if they interpret them faster,
Yeah, like
but I mean, like trying to catch a fly is almost impossible. And they have compound eyes as well. So is that the fact that the compound eyes perceive that somebody's trying to swat them or catch them or something like that, and they move faster? Or are they relying more on the Ocelli because they see a hand sweeping over, causing a shadow to move across the edge of the fly then? I don't know. Could be a combination of the two. Single lens eyes, capable of focusing light on a layer of photoreceptors as it enters through a small opening. And again, we're still in invertebrates. Adjust to compensate for varying light intensities. Focus by moving the lens back and forth. This is found in spiders. You find it in a lot of spiders, but especially well developed in jumping spiders. Okay? Jumping spiders, if you've ever been out in the yard, you can see these things. They move very quickly and, well, they jump. But they have great reflexes. Most spiders you see kind of move slowly. Maybe they're dropping from a web they're releasing, some spider silk. Or maybe they're just sitting in the web waiting for the prey to come to them. They may not have as well developed of an eye. But for something that moves and moves relatively quickly for an invertebrate, then yeah, it's going to have really well developed single lens eyes. So, jumping spider. Yeah.
I heard that spiders, their silk is stronger than steel.
Tensile strength is stronger than steel. Yeah. So, I mean, if you just look at a strand, you take a strand and pull it apart. But if you wove steel that thin and try to pull it apart, you can do that too. The silk can be stronger. So if you take that silk, you compress it down, yeah, you could have something lighter and stronger than steel.
Did they ever try that?
I don't know. I'm sure they have. There's a whole field of study. Bio what? Say it again. Mimetics? That might be it. I can't remember. Or they look at nature and they see what nature does and figure, well, they've been doing it longer, so they're probably doing it better, and they try and mimic what's done in nature. I think the San Diego Zoo sponsors a conference every now and then. I've seen this advertised in the area. Pretty interesting. So jumping spiders, because they move very rapidly, because they're predatory, have very well developed sense of vision based on the single lens eye. So let's start with the vertebrate single lens eye. Iris adjusts to regulate the amount of light entering the pupil. I'm not going to worry about the anatomy of the eye right now. We'll cover that in the lab. And yes, you will need to know it for lab, but not for lecture. Aquatic vertebrates focus by moving the lens back and forth. Terrestrial vertebrates focus by altering the shape of the lens, known as accommodation. Okay, just like with sound, light travels differently through different media. It's going to travel differently through air than it does through water. So you have to have different ways of perceiving. You've probably seen fish before. This eye is probably moving back and forth, up, down, while it's looking for something. Okay? The whole lens will move. Our eyes don't move that way, okay? Bald eagle. Some of these birds have the best vision in the vertebrate kingdom. They can see great distances with very much detail, much better than us. But their eyes don't move like the fish eye. The whole thing doesn't move. The shape of the lens moves, which would be almost imperceptible to us. And that shifting of the lens is what's known as accommodation. You accommodate trying to focus by the amount of light available, by trying to focus on a specific image accommodation. Pick the arrow one because it can see underwater and above water too. It likes to pick off insects on trees that's got branches hanging over water. It can actually leap out of water a good 3 or 4 feet, and they can be about a meter long. So it can jump almost the entire body out of the water. So their eyes are very good for perceiving things in and out of water. Color vision, found in all vertebrates but not all species. So, we can see color vision occurring in fish, amphibians, reptiles, birds, and mammals. Most fish, amphibians, reptiles, and birds have strong color vision. I picked things that were very brightly colored. This is the mandarin goby, it's a saltwater fish. You don't produce these bright colors if somebody of your species can't see them. It would be a waste of time. Tree frog, this is a chameleon, and then of course the peacock. I left mammals off. Humans and other primates are among the minority of mammals with color vision. If you think about most mammals, think about most mammals. Black and white, tans, shades of gray, not much color. Any kind of coloration they have is mostly for camouflage, for blending into their environment, not for attracting mates or anything like that. Tigers, bright orange. But if you're red-green colorblind, like most mammals are, then what happens is your coloration blends into your surroundings very nicely. Because the red and the green just kind of blend together. You don't see it, the orange doesn't stand out. If you think about things like jaguars, leopards, cheetahs, yellow with black spots. That sounds like that would be bright coloration, but it helps them to blend into their environment. Most mammals do not have a lot of color because they don't see in color. Primates being one of the few exceptions. Rods and cones. Rods are sensitive to light but can't distinguish colors. You will find more rods in nocturnal animals, animals that are active at night. Next time you're out at night, look around. You're not going to see a lot of coloration. You see black, white, shades of gray. So, if you're only going to be active at night, why have color vision? It would be a waste. Ditch the cones. Develop more rods to allow you to see better at night, like in the case of this raccoon, which I don't know what he's up to, but it looks like no good. Hanging out on the street looking for trouble. Now, humans, on the other hand, a long time ago decided, let's not be fleshy out at night. Let's be active during the day. Let's be diurnal. So cones became more numerous in diurnal mammals. Less sensitive to light, can distinguish colors, have about 6 million per eye, with the rods 125 million per eye. Humans, we have about 150,000 cones per millimeter squared, equals about 1 million. So now you see why they see better than us. So, on the back side of the eye, we will dissect this and you can see about where it is. You won't see individual rods and cones, they're microscopic. But you see the rod is reddish, the cone is yellow. And in this case, you're going to see more cones than rods, so this organism is probably diurnal. When I used to work at the Cincinnati Zoo, it was called Nocturnal Adventures, an overnight program. Kids actually got to come in and spend the night at the zoo, and so we would make use of that. They'd come in, we'd give them a brief introduction, we got to know the group, we would explain a few things about what was going to happen through the introduction and everything. We'd take them out on a tour of the zoo, we'd come back, we'd do animal demonstrations, bring animals into the room with them, we'd play games with them. The last thing we did at the end of the night was take them on a night hike. And it wasn't to see anything out because most of the animals were put up. But it was just to get the experience of what it was like I would even have them sit and listen, and then close their eyes and listen. And just the sounds you would hear, because when you look, you don't see much at night. And you're trying to, so you don't pay attention to the sound. But when you close your eyes, you hear so many more sounds. I'm trying to teach them what it was like to be nocturnal. The one funny thing was, we'd go over to the eel, a diurnal species. So I would take my flashlight, shine in their eyes, and get their eyes to glow back, meaning they had some rods on them. Why would something that's diurnal, like an eland or a giant antelope, want to see at night? It's not going to be active at night. Predators, risk of being eaten at night. Lions, leopards, hyenas, all active at night. So even though you're day active, you still have to be on alert at night in order to see this. Yeah, I had a lot of fun with that. One night we were out, and we were by the cat house. I know, one connotation means house of prostitution. That's not where we were. It was a place where we actually had small cats and a few big ones. And we're there because during the summer when the cats were housed outside, the small cats, oh, the odor they would put out, marking their territories, which is really intense. And I would tell them, as you're walking up to this, you smell this. And if you're maybe another cat looking for something, you can tell if this cat's inviting you in to maybe mate, make cubs, whatever. Or if you're a prey species and you smell this, you might want to avoid that area. So you can use your sense of smell here. About the time I'm telling them this, all of a sudden the bushes start to shake. And you turn around and look, and I'm thinking, out loud, stupidly. Let's see, on the other side of that hill, well, that's where we keep our lions and tigers. And all of a sudden the bushes parted, and the biggest possum I ever saw ran out. You could literally hear the screams all over the zoo. I mean, I'm dealing with like 8 to 10-year-old kids. I'm probably scarred for life. We get back on the other side of the zoo where the education center was, and they asked us, "Man, did you hear that scream?" I said, "Yeah, what was that?" So, always got to watch what you say. A couple of pigments. Rhodopsin. Visual pigment found in rods. Rhodopsin, rods, they kind of go together. As rhodopsin absorbs light, it changes shape, making rods unresponsive. Moving from dark to bright light requires several minutes for bleached rods to become responsive again. Okay, like I said before, it comes out in eye shine. You see it in night active animals or animals watching out for night active animals. It's dark out. Maybe let's say you're in bed and something wakes you up and you panic and you turn on a light and all of a sudden you're blind. You can't see anything. It's because you've bleached out the rods. So what little you can see at night, all of a sudden is incredibly ineffective because you just bleached them out. And now your cones are starting to wake up and think, oh, my turn. And trying to see what there is in the room that made the noise, whatever woke you up. So you experience this bleaching. It's happened to you before, guarantee it.
So, because how does it work when you close your eyes before you turn the light on? So that way your eyes are better adjusted.
It could be, yeah, start to adjust it for night.
So like, well, because I know like when you're in a dark room and you turn the light on, you're supposed to close your eyes like before turning the light on, so that way you don't get blinded.
Right.
Is that because our eyelids like kind of transparent so light goes through?
You can sense light a little bit, but I mean, eventually you're going to have to open your eyes. Maybe you're going to squint and open it one at a time instead of, having your eyes wide open all of a sudden the light coming on. It's just a transitional period that you can adjust to a little bit easier. You're still going to bleach things a little bit, and it's just going to happen. Yeah.
Can that have an effect, like say, or like a delayed effect? Like say if you passed out and then you were like waking back up, and you kind of see like white light.
I don't think that has anything to do with that.
Okay.
I think it's more of something just going on in your brain. If you maybe you stand up too fast and you get a little bit of a head rush.
Yeah.
I think it's more along those lines when you pass out than something like this. Bleaching effect.
Okay.
A little bit. I've never done that. But I mean, that's how you get the eye shine. You can shine that in their eyes, and it will reflect off the back, off the rhodopsin, and emit out. The deal is with rods, they take what little bit of light is available and magnify can see so well at night, and why we can't, because we have more cones than rods. We also have photops, which is another pigment. It's actually 3 visual pigments found in cones: red, blue, and green, RBG. Overlapping absorption spectra allow for intermediate hues. So with these 3 basic colors, as they overlap each other, whether red overlaps blue, blue overlaps green, green overlaps red, or they all overlap each other, you get a pretty broad spectrum of colors. We're talking about bigger than the 64 Crayola crayon box. You see a lot of different colors. Color blindness occurs with the deficiency or absence of one or more types of photopsin. Usually people that are colorblind or animals that are colorblind can see some colors because they're only missing one shades of gray. And of course, dogs are colorblind. I like the movie Up. At the end of the movie, they're all sitting there counting the color of cars go by, red and blue. And you hear the dog say, "Gray." It's a gray car. That's all I thought. Yeah.
Is there a way to fix like being colorblind?
Not that I know of. Not that I know of. Basically, you just adapt. I mean, if you've been colorblind all your life, you really don't know what you're missing out on. If you haven't seen it before, you probably, your brain has probably already adapted to it to some degree. Okay, last thing we'll talk about today, we're not taking a break, but we're going to finish probably in the next 15-20 minutes. Animal skeletons. There are 3 main functions of a skeleton: support, protection, and movement. Support, this is what allows us to stand upright. Because we've got a skeleton system designed to keep us up. And if you're a quadruped, 4-legged, then it keeps you horizontal to the ground. But it allows you to stand up, fighting the effects of gravity. That's why I love this picture. I mean, you see the largest land animal standing on its hind legs, reaching above itself. I mean, if that's not a good indication of support in the skeleton, I don't know what it is. Protection. You've got your skull that protects your brain, your sternum that protects your heart and some of the other things in there. Your rib cage protects your lungs, a little bit of the liver. Movement. I mentioned this before when we talked about muscles. Your muscles need a fixed point, something around which to move. They need leverage. And so what happens is they attach to your skeleton, which just gives them a fixed point that allows you to do things like this. Okay, that's 2 different muscles working against each other, allowing you to move. Physical support on land, body posture, position of legs to make body is an important structural feature in birds and mammals. Okay. We talked about this with mammals. Remember, our legs are underneath us, not out to the side. Not like reptiles. They are underneath. And so it gives us this pendulum motion, allows us to move a little bit more efficiently, a little bit faster. Birds the same way. When they started making the Jurassic Park movies and the Walking with Dinosaur documentaries, they started to plug in everything about dinosaurs they thought they knew. And the computers kept coming back, it does not compute. Got to look at it a different way. This isn't working. Dinosaurs could not have moved this way. And so they started looking at things like birds and like other mammals like elephants. And they realized there's no way the dinosaurs could have walked like we always thought they did, standing upright, dragging their tails behind them, with their head, way up. They had to be more evenly positioned over their legs in order to move more efficiently. So body posture is very important back then and now. We're going to go over 3 different types of skeleton. The first one is the hydrostatic skeleton. It's got hydro in the front of it, so you know it has to do something with water or fluid. Fluid held under pressure in a closed body compartment. Muscles change shape in fluid-filled compartments. Found in cnidarians, flatworms, nematodes, and annelids, the earthworms. Take an earthworm for a second. It's got muscles all along its body. It's that peristaltic movement, this wave-like contraction. Those muscles couldn't work if they didn't have something to work around. So if you're talking an invertebrate, it has to have something to use to give those muscles leverage to allow them to And so what it does is it takes sebum, basically, this compartment, fills it with fluid, and that gives it enough pressure to allow the muscles to work. So if you don't have a skeleton, you use fluid, kind of like hydraulic pressure. A fluid-filled compartment that allows these organisms to move. Next up, the exoskeleton. Hard encasement deposited on the surface of an organism. In arthropods, opposed to chitin, exoskeleton periodically shed for growth, also found in mollusks and arthropods. Okay, you've got something like a beetle. It starts to grow. It gets so big, and this hardened exoskeleton now is no longer big enough for it. It's getting kind of tight. So it sheds that, it molts, and then it grows a little bit bigger. And when that exoskeleton gets too tight, it sheds and molts again. And this keeps occurring until it gets to adulthood. Very rapid when they're young, but as they mature, as they get older, it doesn't happen as frequently. Remember, this ecdysis is a very high energy process. It costs a lot of energy to do this. Now, just like with us, they have muscle attached to their skeleton so they can move. It's just that unlike us, we've got our soft gooey parts on the outside, the hardened skeleton on the inside. They've got their hardened skeleton on the outside and all the soft gooey parts on the inside. But they still use their skeleton, even though it's an exoskeleton, for muscle attachment. Like we said, opposed to chitin, just like these cell walls of fungi. And mollusks have an exoskeleton. Think of things like clams, oysters, mussels. You can count the shells of certain organisms like the nautilus, snails. So we've had the hydrostatic skeleton, we've had the exo or outer skeleton. The only thing that's left is unlike ours, the endoskeleton. Hard supporting elements buried deep within soft tissues. Now, to bend your mind a little bit, not thinking that it's just vertebrates that have something like this. They're the biggest group that have most successfully utilized the endoskeleton. But sponges are reinforced with spicules. That gives them structure and support. Technically, that's a type of endoskeleton. Echinoderms, reinforced with bony plates. You guys dissected the starfish. You know that it was a little bit difficult to cut through. It wasn't just like cutting through a piece of skin. It was hard. It gave a little bit of resistance. That's their endoskeleton. And of course, chordates, reinforced with cartilage and or bone. I don't know if I'll do it with you guys. I'll tell you now. Maybe I will. Putting the shark skeleton on the lab exam. It is entirely made of cartilage. They're cartilaginous fish. And it's funny because people see this and they don't think it's a real skeleton. It's made of cartilage. By definition, an endoskeleton or a vertebrate can be composed of cartilage, bone, or a combination of the two. We happen to be a combination of the two. But remember, we've got cartilage all throughout our body. We've got it in our nose and our ears for shape and support. We've got it in between all of our bones so they can glide across each other, not always grinding across each other. We have it in the intervertebral discs between the vertebrae and our back. Acts as a shock absorber there. Just as the sharks have an entire skeleton composed of cartilage.
Is that one in water?
Yes.
Okay.
So, technically, sponges, echinoderms are also members of the endoskeleton family, just like us. We say chordates, more than just vertebrates here, because many chordates have at least a rod of cartilage running down their spine, and that counts too. Muscles work in antagonistic pairs in order for body parts to move in opposite directions. three muscle types: skeletal, cardiac, smooth. We've covered these in lab, so you have an idea of what they are. To flex the arm, the biceps contract, triceps relax. To extend the arm, the biceps relax, the triceps. Triceps and biceps work against each other, and they're right above the elbow joint. And what they do, not only do they allow the arm to move, they make sure the other muscle doesn't hyperextend or overextend. Sometimes it works, sometimes it doesn't, depending upon how much force you exert on your muscles. Usually when you talk about an antagonist, it's the bad guy in the movie as opposed to the protagonist. But in this case, we're not saying one muscle is the bad guy, the other is the good guy, or vice versa. We just say they work against each other so they don't hyperextend or damage the joint. Again, the three muscle types: skeletal, striated and voluntary; cardiac, striated and involuntary; smooth, unstriated or non-striated, and involuntary.
Well, all muscles have like a hair or
No. Some do, yeah. I'm not exactly sure what triggers it, but the nerve in the muscle keeps firing and causes the muscle to contract and relax. So why that nerve does that, I don't know. It could be the muscle could be overused, and it just causes that to happen, because sometimes when you put a lot of force on a muscle, it causes the muscle to contract, and sometimes after you release that force, you still feel the muscle kind of shiver, shake a little bit. That's because in the muscle's makeup, it's still thinking it's contracting, and you reach what they call tetany, and it just repeatedly contracting. Something triggers something like that to happen in muscle spasms. What exactly, I'm not sure. It could be like an imbalance of electrolytes or something like that, or maybe sodium and potassium levels are off.
So is that the same for like a trolley horse? Where it's really
That just contracts and doesn't want to let go. For some reason, your muscle just contracts, just holds on. Muscle spasms contract and release, so you can actually see the muscle spasm. So for some reason, it's all nervous system related, because that's what causes muscles to relax. But why it does with the Charlie horse and with the muscle spasm. A couple of fun to know facts about muscles that you will need to know. Arthropod, vertebrate, skeletal, and smooth muscles are nearly identical. Kind of crazy. Something that primitive has muscles that are almost just like ours. Insect flight muscles can be faster than action potentials arrive. Ooh, that kind of ties into something we talked about last time. Remember, action potentials move through the nervous system like a wave. But if you're something like a dragonfly or some other insect whose flight muscles move very rapidly, they're probably moving faster than those waves can be moved throughout the nervous system of the insect. And this is where something like electrical junctions occur more so than neurotransmitters. Instead of chemical synapses, you have electrical synapses to keep up with that. Clamp adductor muscles can remain contracted with low energy consumption for up to a month. And you're thinking, so what? Well, we looked at both starfish and we looked at both and clams. Starfish will come up to a clam and put its arms on either side. I'm not going that far. Just relax. And so what happens is they take these tiny little tube feet, their pressure, and start pulling that clam apart. Now, if the clam didn't have strong adductor muscles, every time the starfish would come up to it, it would be dead. But if it can take those adductor muscles, the anterior and the posterior, and hold them together really tight, as you can see, it was difficult to cut through. You had to cut through the muscles in order to open the shell. Then it's going to make it harder for that starfish to pull apart. So who's going to win out? I guess it depends on who's got more patience. The starfish can withstand that. Up to a month. Crazy. Okay, Locomotion. Last 3 or 4 slides. First up, flying. Well, we've got flying, we've got swimming, and then we've got terrestrial locomotion. Gravity is a major problem that wings must overcome with lift. Shape of wings is significant for the type of flight. If you look at the hummingbird, its wings are a very basic design. It's going to beat very rapidly, and it maneuvers much like a helicopter. If you look at the peregrine falcon, its wings are actually more like a fighter jet. They can extend out like this, but in a dive where it's trying to pick up speed, it can actually fold the wings back. They don't use them much anymore, as much. The F-14 Tomcat can do that, where they sweep the wings back for greater speed. Here, I'm not sure what this is. It's just an albatross or a frigate bird. I think it's an albatross.
Yeah, that's what it looks like.
Has very long, thin wings, not a lot of flapping. It tends to spend most of its time just coasting on the coastal breezes,
Like gliding.
ocean breezes. Do what?
Gliding.
Yes, more gliding. So, shape of the wings has everything to do with the type of flight. A wing like this would not work very well for the hummingbird. Adaptations exist to reduce body mass. We've talked about all this before. How they have feathers, wings, bones that are air-filled, not hollow, but pneumatic. They don't have teeth. They've reduced a lot of their internal organs to give them more lift, less weight. And of course, fusiform shape for aerodynamics. We've talked about this round. What's the fusiform shape?
So tapered at both ends.
Tapered at both ends. So the biggest problem they have is gravity. Trying to be able to take off without gravity pulling you back down. Swimming is actually the most efficient mode of locomotion. Friction is a major problem that, again, the fusiform design must overcome with hydrodynamics. Water is tensive in air, which creates friction, but it provides buoyancy to cope with gravity. So, gravity is not the biggest problem. Friction is here. Diverse methods of swimming allow animals to overcome friction. You've got something like an eel that can fly through the water much like a snake does on land. You've got something like the ray that actually flies underwater. And then you've got something like the bluefin tuna that moves very quickly, really taking a lot of advantage of their fusiform design. Not only are they tapered at both ends, but side to side they're very, very flat. So the 3 types, flying, swimming, and terrestrial locomotion, swimming is the most efficient. And their biggest problem is friction. Again, with flying, the biggest problem was gravity. But that buoyancy is what allows whales to get as big as they do. They could never survive on land. They're too massive for that. The biggest whales are bigger than the biggest dinosaurs. But being in the water helps to support their weight. It doesn't crush them like those whales that get beached. And so it allows them to get really, really big. Like I said, we've got flying, we've got swimming, we've got one flat. And that would be terrestrial locomotion, whether it's walking, running, hopping, whatever. This is the least efficient. Friction is a minor problem because air is less dense than water. However, gravity is a major problem due to the animal's necessity to support itself. Larger animals require more energy for locomotion. Remember, we talked about the mouse and the elephant. Gram per gram, mice burn more energy, but there's just a lot more grams for the elephant. So overall,